Okay. Hi, happy Dasra. Uh, I'm Nandika. I'm going to be conducting this session. And welcome to problem solving session for week 12 for experimental biotechnology. So, as you must have seen, this week there is basically just a summary of what we've discussed in the previous weeks. So, I thought we can go through some questions together. If you have any doubts, please feel free to ask me. If you have any other doubts related to the content, you can also ask me. But yeah, till then we can just solve some questions. Yeah, happy Dasara. Okay. So this is just a selection of MCQs based on what we've seen so far. This, as you can probably tell, is an agarose gel in which they have probably done some random PCR on genomic DNA for some organisms A, B, C, and D. And because all of these organisms have different genomic DNA, they are asking, each of them will give a different banding pattern, right? And that banding pattern will be characteristic for the organism. So now the question is, which organism has the most DNA in common with organism A? So if we take this well as a reference, okay, then we have to see which of the organisms B, C, and D are the most similar to A. So if you look at this first band, it's there in B and it's there in D. However, all three of these have this other band which is not there in A. That's okay. Let's see the others. Now A has these two bands, present in B, present in D. This band is present in D. And this band which is absent everywhere. So if we look at how many bands are there in common, for B there are 3 bands which are common with A. For C there is 1 band. And for D there is 1, 2, 3, 4 bands which are common. So the organism which has the most DNA in common with A will be organism D. Okay. This question is, when the pH of a protein is lowered, dash will happen. Okay. We remember that the protein is made up of amino acids, and each amino acid will have, well, each amino acid has 3 ionizable groups, right? You will have the amino group, right? You will have the carbonyl group, and you have the R group, and sometimes the R group will also be ionizable, right? And the overall charge on the protein is basically coming from what is the charge on the R group and what is the charge on these two ends. So at pH 7, an amino acid will look like this, where the amino group is protonated, right? And sorry, this will lose this hydrogen and become COO minus at pH 7. However, as we decrease the pH, the hydrogen ion concentration is increasing, right? As we go to lower and lower pH, the hydrogen ion concentration will increase, and therefore this group, the equilibrium will shift further towards this being protonated. Okay, so as we move to lower pH, basically the negative charge on the protein is reducing, or rather negatively charged groups will be neutralized, right? Because equilibrium will shift towards the hydrogen associating here. So under neutral pH, negatively charged groups will be neutralized. A protein is neutral at isoelectric point. This concept has been come up many times in the course. Isoelectric point, or pi of the protein, is the point at which net charge of the protein is 0. Okay? This does not mean that the protein has 0 charges on it. It just means that the number of positive and negative charges on the protein is equal. Okay? So the overall charge on the protein is neutral. Right? So this is true. Okay. Which of the following is used as an ion exchange resin? Ethanol, cellulose, starch, and collagen. Now, remember, when we talk about a resin for chromatography, it should have a couple of properties. Okay, it should be stable. Okay, it is usually a solid phase. Okay, and it should be easy for us to couple the groups which will react with our molecules of interest. So when we look at ion exchange resins, the only one of these that is commonly used is cellulose. In ion exchange chromatography, proteins bound to the resin can be displaced by increasing the dash. Right? Remember, in ion exchange chromatography, we have a resin, it has a certain charge on it. Let us assume our resin has a positive charge. Okay? Then, how the chromatography works is when we pass our protein solution, all the molecules with opposite charge are going to get stuck to the resin. Alright? So all the molecules with negative charge are going to get stuck here, while everything with positive charge and everything which is neutral is going to flow out of the column. Now, after passing this flow through, our target protein is stuck on the column. But basically, to elute this protein from the column, we have to break this interaction. Now, there are two ways in which we can break this interaction. One is that we can alter the charge on this protein. Right? So we can change the pH of the buffer. If this protein is no longer carrying a negative charge, then it won't be attracted to the resin anymore. It will be eluted. The other way is by increasing the ionic strength of the surrounding solution. Okay? As the ionic strength increases, okay, it actually disrupts these electrostatic interactions. And so normally when we elute proteins from ion exchange columns, we usually have a gradient with increasing concentrations of a salt like NaCl. And because we are increasing the strength of the ionic buffer, it is disrupting the electrostatic interactions and allowing our protein to elute from the column. Okay. Gel filtration chromatography separates proteins based on their dash. Gel filtration chromatography is also called size exclusion chromatography, okay, or gel permeation chromatography. It is basically making use of a porous matrix, and depending on the size of the protein, it will be able to enter the pore, or it will be too big to enter the pore, and it will just move around the beads. Right? So the bigger protein, because it cannot enter the pore, it will have a shorter path length through the column, so it will elute first. While the smaller proteins, because they can enter the pores, they'll have to travel more distance, right? So they will take more time to elute. So the proteins are being separated based on their effective size. In gel filtration chromatography, the proteins dash. So as we discussed, in gel filtration chromatography, there is no interaction between the protein and the resin as such, right? We're just making use of the pore size to allow the protein to pass through that path or not. So it is not binding to the column in any way. Okay? So the protein is just moving through the column The correct term would be that the proteins are diffusing through the column. Okay. Proteins interacting with specific substances can be separated using dash. As we know, there are many different kinds of chromatography, and which one we select will depend on what kind of interactions our protein has exhibiting. Now, in this question, they are saying that the protein has a specific interaction. Okay, and specific interactions are very good because we can exploit them by putting the interacting partner on the resin. Yeah. We can put the interacting partner on the resin. And so, because this partner is here, when we pass our protein through the column, it can bind to its interacting partner, and so only the protein of our interest will be stuck on the column. And this process is called affinity chromatography, right? Because we're making use of the affinity between the protein and its binding partner. Okay, and we're making use of a very specific interaction. Things like ion exchange chromatography, we're just making use of attraction between opposite charges. So that's a non-specific interaction. Okay. Polyacrylamide gel electrophoresis uses dash to separate proteins. Right? Polyacrylamide gel electrophoresis, if you remember, is when we cast this sort of gel made out of an acrylamide polymer. We load our proteins at the top, and then we apply a voltage difference across this gel. Okay. And then the proteins, we usually run this in SDS, so the SDS imparts a negative charge to our protein. So our proteins are thus migrate towards the positively charged electrode. Right? And so the proteins are separated based on their size due to the influence of the electric field. There is no other difference in pressure, temperature, magnetic field, so on in gel electrophoresis. Okay. Before starting to study a protein, it is usual practice to do dash. Determine the protein's amino acid sequence. Is it to confirm the protein's role in the cell, determine its molecular weight, or purify the protein? So before we start to study a protein, we have to think about what are the purification strategies we can use for the protein. And for that, it is very necessary to know the nature of the protein. Like, how big is it? What will be the charge on it at different pH? Is there any special property in its structure that I can make use of? Is it binding to some other protein? All these things are very useful quantities that we should know before we try to express and purify a protein. And most of this information you can derive from its amino acid sequence. Okay? It is very necessary to know the protein's amino acid sequence if you want to do a specific purification. Okay? Because if you don't even know the size of your protein, it will be very difficult to purify it and work with it in the future. So usually the first step will be to determine its amino acid sequence. Okay? From the amino acid sequence, we can then determine the molecular weight. Once we know how to identify a protein, we can do other things like confirm its role, okay, try to overexpress it, try to purify it, and so on. But the most basic information is we should know its sequence. Okay. This is more in the context of an enzyme, but they're asking what the term specific activity differs from activity in what sense? Basically, what is specific activity? So, activity of an enzyme is usually defined as amount of product produced per unit time or amount of substrate consumed per unit time or something like that. But then what is specific activity? Specific activity is actually activity in a milligram of protein. Okay, this is the activity in a set amount of the protein mixture, and this can help us determine the purity of the protein. So when we start a purification procedure, say we express our enzyme in a cell, we directly take the cell lysate. Okay, in the cell lysate, there will be so many other proteins apart from the protein of our interest. So if we take 1 mg of that cell lysate and measure its activity, only a very small part of that is our protein. Right? So the specific activity of that will be low. But as we purify our protein more and more, the amount of that 1 mg, which is containing our enzyme of interest, is going to increase. And so the activity of that 1 mg is going to increase. So specific activity is basically a measure of the purity of your protein. Okay? In a successful purification protocol, it may be expected that dash, as we just discussed, uh, one measure we use of purification is that the specific activity of our product should increase. Okay. The total activity is not going to change. In fact, total activity might reduce slightly because we are not changing the amount of the protein that is present. Right? We're just purifying it. And during the purification procedure, we will probably lose some amount of protein, so it's possible that the total activity will reduce. Okay, but if the purity is increasing, then specific activity will also increase. Okay. When you measure the amount of protein in a solution by UV absorbance, you are detecting the UV absorbance of predominantly which amino acid. So the most common method of protein quantification is to measure absorbance at 280 nanometer. Okay. And the absorbance at 280 nanometers is basically contributed to by two major amino acid residues, tryptophan and tyrosine, but the major contributor to this is tryptophan. Okay. Basically, the aromatic amino acid residues absorb in the UV range. Phenylalanine also absorbs, but it absorbs at a slightly shorter wavelength. Okay, if we look at a diagram, say this is 250, this is 280. So phenylalanine will absorb something like this, around 250, 260, whereas tryptophan and tyrosine have absorbance near 280. So this is also something to keep in mind, that if you have a protein without a tryptophan residue, okay, it is possible you may not be able to detect it by absorbance at 280 nanometers. Which of the following reagents is used to release integral membrane proteins? So, as I'm sure you know, an integral membrane protein is a protein which is going to be embedded in the membrane, like this. And when we want to purify these integral membrane proteins, we have to remember that this region of the protein is all hydrophobic, right? Because it is inserted inside the membrane. So if we just take it out of the membrane, okay, it is going to be exposed to the aqueous environment, right? And as we know, because of hydrophobic interactions, protein will then collapse, right? Because it will try to take all the hydrophobic regions to the core and expose the hydrophilic region to the environment. So the protein will collapse. So therefore, when we extract these integral membrane proteins, it is very necessary that we maintain some sort of hydrophobic environment in this region. So it is usually extracted using a detergent. Detergents, I'm sure you know, are amphiphilic molecules. They have a hydrophilic head and a hydrophobic tail, and they can organize to form these spherical regions called micelles, right? Where the hydrophilic heads are pointing outside and the hydrophobic tails are pointing inside. And what we can do is add detergents to our membrane, and it will stabilize the membrane proteins. Okay. Now, in this list, actually only one of these solutions is a detergent. Okay. Urea is a chaotropic reagent. Okay, it is going to denature your protein. It is not a detergent. DMSO is an organic solvent. Cyanogen bromide is used for things like coupling reactions and protein cleavage. So Triton X is actually the only detergent in this list, but it is used to release integral membrane proteins. The salting out process involves precipitation of proteins. In what way? Salting out process is usually the addition of ammonium sulfate to the point where the solubility of the protein is reduced and it will fall out of the solution. Right? So protein will precipitate from a solution at a specific concentration of ammonium sulfate. Okay? There is a series of which ions is best for salting out, and ammonium and sulfate ions are actually very effective at this, which is why we use this compound. Finally, a project student added ammonium sulfate to a tube containing crude extract of a protein, then centrifuged the solution. What is the student probably trying to do? Like we just discussed, when we see ammonium sulfate in a protein purification protocol, it is going to act to precipitate our protein. Right? So, of these options, does change the pH of the buffer solution? Probably not. Hydrolyze the proteins into the constituent amino acids? Ammonium sulfate is not going to do this. Denature the protein of interest? No. Ammonium sulfate is going to selectively precipitate our particular protein. At certain concentrations, different proteins precipitate. Okay. Which of these following techniques is not used to concentrate proteins? Okay. Binding and release, ultrafiltration, precipitation, and dialysis. Okay. So, to answer this question, we should know what each of these techniques is doing. So precipitation, you can see that obviously our protein is going to fall out of the solution, and then we can re-dissolve it in a smaller amount of our solution, right? So we can use this to concentrate our protein. Ultrafiltration. So this is you get these sort of centrifugal filters, okay, which have a specific cutoff. Okay, and because of this, the buffer passes through the filter while our protein remains in the filter, okay, because it's above the molecular weight cutoff of the filter. So this is commonly used to concentrate proteins. Binding and release, same deal, right? When you do affinity chromatography, think about it. When you do affinity chromatography, you will pass a large amount of volume through this column, right? But you'll elute it in a small amount of liquid. So this is also going to concentrate your protein. But when we do dialysis, right, what is happening is we have a dialysis bag, right? It's clipped on both ends like this. And we're going to keep it in a beaker containing our buffer. Well, I'm sure you're familiar with the principle of osmosis. So what is happening here is that a solute concentration is higher, and here the solute concentration is lower. So what is going to happen is to equalize these concentrations. Since our protein is stuck in the bag, right? We use a cutoff which is less than the molecular weight of our protein. We don't want our protein to go into the buffer. So what is going to happen is that solvent is going to come into our dialysis bag, okay, and increase the volume of the solution that is kept over there. Yeah, so if you've ever done a dialysis, you will notice that the bag swells up by the end of dialysis. So dialysis is not concentrating our protein, okay? In fact, it is diluting our protein. Okay. The liquid emerging from the bottom of a chromatography column is called the dash. Okay. So in very technical terms, the liquid emerging from a chromatography column is called the eluate. Okay. No one uses this term in actual work, but it is actually called the eluate. Which property is the basis of separation of proteins by SEC? Okay, the clue is in the question itself. Because it is size exclusion chromatography, it is separated on the basis of the protein's molecular mass. Indicate which one of the following types of column can be used to estimate relative molecular mass. So this is actually something we can do using SEC, right? Since different proteins take different time to travel through the column. Okay? Right? So my smaller protein is going to take a much longer path, so it's going to take much longer to travel to the column. So if I have something, okay, I have elution time on the y-axis like this. If I have something of known molecular mass, and I know this is eluting here, and I know, say, this is 100 kDa. Okay. If I have any unknown protein which is traveling through the column, and it elutes over here, so automatically I will know that its mass is less than 100 kDa, right? Because it is taking more time to come out of the column. Okay. Of course, you can get a far better estimate of approximate molecular mass if you have like multiple known standards. Okay. What is true for size exclusion chromatography is that elution volume is proportional to the log of the molecular mass. Okay. So what you can do is plot molecular mass versus elution volume. Okay. Or rather log molecular mass versus elution volume. Find the equation of this straight line. Okay, and then you can use this to predict the approximate mass of any other protein that you are passing through the same column. Okay. So this is one of the benefits of analytical size exclusion chromatography. You can estimate the molecular mass of any unknown protein. Okay. When separating proteins using size exclusion chromatography, the first protein to be eluted will be dash. So as we discussed, the largest proteins will not be able to enter the pores of the matrix in size exclusion chromatography. So the larger the protein, the more quickly it will elute from the column. So the first protein to be eluted will be the largest protein. Okay. Affinity chromatography involves dash. Clue is in the name again. Affinity involves specific binding of a protein constituent to another molecule. All of these others, it can be a protein-protein interaction, it can be a protein-carbohydrate interaction. But what is important is that it is a specific interaction. Okay. What properties of a protein does hydrophobic interaction chromatography exploit for purification? Okay, this is another type of chromatography we've discussed in this course, wherein our column itself is hydrophobic. Right? And as a result of that, the greater the hydrophobicity of the proteins passing through the column, the more likely they will be to bind with the matrix. So what we're exploiting is hydrophobic amino acids on the protein surface because it needs to interact with a resin. Okay. What is the starting point of selection for a suitable ion exchange matrix for purification of a recombinant protein? So how do we know which ion exchange matrix we should choose? Right. So basically what we should happen is that our ion exchange matrix should have the opposite charge as our protein. Okay. So prediction of isoelectric point from the amino acid sequence. Okay, this is very important. Only once we know the isoelectric point will we know what pH the protein will have in our buffer. Right? So if my protein has pI of 5, but pH of my buffer is 7. Okay, so here I know that the charge is 0. The question is what charge is it carrying in pH in the pH 7 buffer? pH 7 buffer, it will carry a negative charge. Okay? If I wanted to carry a positive charge instead, what pH buffer should I use? Right? I know that the pi is 5, so I know if I wanted to carry a positive charge, I have to go for pH which is lower than 5. So if it needs to be positively charged, I will have to make a buffer with pH less than 5. So something like pH 3. Okay. Test protein binding to IX matrix at range of pH and salt concentrations. This we can do, but this is not where we would start. Okay. This you will start if you don't know anything about the protein sequence or anything. Test protein binding to selection of anion and cation exchange matrices. Again, if we don't know anything about the protein properties, we will do this. Okay, pass a sample through preparative column and elute with a salt gradient. This is part of protein purification, but the most basic step, if we know the amino acid sequence of a protein, and it is a recombinant protein, right, which means that we have cloned it and we are overexpressing it in the organism, so we already have some knowledge about the protein. So since we know the amino acid sequence, the best place to start is to predict the isoelectric point. Then we can predict what the charge on the protein is going to be, and then we can select the appropriate ion exchange resin. Which functional group can be found on an anion exchanger? Okay. Just a quick revision. Anion exchangers and cation exchangers. Remember, it is called an anion exchanger, right? Which means that it exchanges anions, right? So the group which is going to bind to it will be negatively charged because anions are going to bind to it. So the resin itself, right? I have some negatively charged anionic compounds which are going to bind to my resin. So my resin should have a positive charge. Okay? So anion exchangers will carry a positive charge. So the group which is usually used as an anion exchanger is diethyl aminoethyl. Okay, you may know it as DEAE. So very common anion exchanger. Okay. DEAE is just the short form of this. Okay. You can see in this aminoethyl, right? So this is the group that is going to carry the positive charge, where our anions are going to bind. Okay. The opposite question, which functional groups can be found in a cation exchanger? Same deal. Remember, the cation exchanger has to bind cations, which are positively charged compounds. So the matrix itself should be negatively charged. There are actually two groups in this list which are cation exchangers. Both carboxymethyl and sulfopropyl are cation exchange resins. Okay, these are both negatively charged resins that can bind cations. In what order would the amino acids E, H, I, K be eluted from a DAEE cellulose ion exchange resin by a buffer at pH 6? Okay. So, to solve a question like this, your concepts about ion exchange chromatography need to be very clear. So, you need to know what is DAEE exchange resin. They've already given anion exchanger by a buffer at pH 6. So, remember the order in which they will elute, right, will be in the order of weakest interaction to strongest interaction, right? Because if it is very strongly bound to a column, it is going to take more time to elute. Now the question is, since it's the anion exchanger, we should think about which of these amino acids will carry a negative charge at this pH. Okay. So if we look at these amino acids, isoleucine does not have an ionizable R group. Okay. So its charge at pH 6 will be uh, it will be slightly positive. Okay, let's say neutral. Okay, for ease of discussion, we'll say isoleucine is going to be neutral at this pH. Lysine has an ionizable side group. Okay, it has an amino group which is going to ionize, so it is going to be an H3 plus. Okay, at pH 6. So a lysine is going to carry a strong positive charge at this pH. Okay, pK of this is somewhere around 10, 11, 12. Okay, this is not going to dissociate at pH 6. Histidine is an interesting amino acid. Uh, it has an ionizable side group with a pK of around 6. Okay, but in interest of discussion, this is also going to be almost neutral, but it may carry a slight negative charge. Okay, because some of the side group will dissociate at pH 6. Glutamate, okay, carries this ionizable side group. Okay, this will have a pKa of 3 to 4. Right? I think glutamate is around 3, aspartate is nearer to 4. So once the pH goes above 4, what is going to happen is this hydrogen is going to dissociate and this will become COO minus. So glutamate is going to carry a strong negative charge at pH 6. Okay? Which means that glutamate is going to bind very strongly to this resin. Histidine is going to bind slightly weak. Okay, or isoleucine is going to be mostly neutral, but it is a hydrophobic amino acid, so it may have some interaction, some level of interaction with this with the resin. And the lysine is not going to interact with the resin because resin is positively charged. Lysine is positively charged, so it is not going to interact with the resin at all. So this will be the first to elute. This will be the fourth to elute. These two are kind of interchangeable. So of these options, D is the most appropriate answer, right? Because lysine will elute first and glutamate will elute last. In anion exchange chromatography, which of these is true? In anion exchange, remember, the protein is negative, the column is positive. So the column contains positively charged beads to which negatively charged proteins bind. The use of the hormone insulin to isolate its receptor is an example of dash. Now here we're using specific interactions between insulin and the insulin receptor. So this is an example of affinity chromatography. A mixture of two proteins which bear similar charges and relative molecular masses 10,000 and 18,000 are respectively applied to a gel filtration column. What will happen? So as we know, in a gel filtration column, proteins will elute in decreasing order of their molecular mass. So first this 18,000 Dalton protein is going to elute, followed by this 10,000 Dalton protein. Okay, so what is going to happen is protein of MR 18,000 will elute first. Since this is a gel filtration column, there are no functional groups attached to the matrix, right? We're just separating on the basis of size. Okay. Mixture of 4 proteins A, B, C, D is applied to a size exclusion column. Here we're given information about the molecular mass. Which protein will elute first? Okay. Let's go one step further. We'll discuss the elution order. Okay. Since we already know it is going to elute in decreasing order of molecular mass, so the biggest protein here is C. So this is going to come first. After C, we have A. So this is going to come second. Now B and D have very similar molecular mass. Okay, this is 25,000, this is 23,000. So ideally, B will come third, D will come fourth, but it depends on the resolution of your column. In a lot of columns, it is possible that you will not be able to separate these two. Okay, the molecular mass is too similar. So B and D might elute together. But the protein which is going to elute first is C, because it is the biggest. We have a small volume of a solution of buffer salts in a protein with MR greater than 10,000. Which of the following chromatographic techniques would be the best choice to separate the protein from the salts? So here we see that the protein has a fairly high molecular weight. Okay, this is more than 10 kDa. So we can actually apply size exclusion chromatography to this. Size exclusion chromatography with a low molecular weight cutoff is also called desalting. Okay? Because what is going to happen is the buffer ions have to pass through all the pores. Okay, if you have a matrix with very small pores, the ions have to pass through all the pores, but your protein is excluded from the matrix and your protein is actually going to elute in the void volume. Okay? So it is going to be separated from the salt. In a protein purification, starting material contained 200 mg and 100 units activity. Okay, we'll just try to find specific activity, which is activity divided by weight, right? Which is 5, whatever, units per mg. The purified material contained 2 mg protein and 800 units of activity. So specific activity for this, 800 units upon 2, which is going to be 400 units per mg. What was the yield percent recovery of activity? Oh, okay. So here they have not actually asked for specific activity at all. Okay. Here they're just asking how much of the activity has been retained. So all of this is actually does not matter for this question. They're just asking in terms of total activity how much protein is recovered. So obviously activity is directly proportional to the amount of protein which is present. So here they've already given us the total activity. So the starting amount, it doesn't matter how much the weight is, it had 1000 units. And after purification, we're left with 800 units. So the yield in terms of activity will be the final activity divided by current activity in 200, which is going to be 80% yield. Right? If it was 100% yield, we would have retained the 1000 units, but we don't. We had some loss. Okay. It's the same scenario, but this time they have asked for specific activity. So they're asking specific activity of the purified material. As you can see, purified material is containing 800 units in 2 mg. So the specific activity will be 800 by 2, which is 400 units per mg. Okay. In the same scenario, what is the relative degree of purification? Okay. So starting specific activity was 1000 by 200, which was 5. The purified specific activity was 400. The relative degree of purification, if we go for fold purification, will be the final specific activity divided by the starting specific activity, which will be 80, right? Yeah. So we have 80-fold purification of the protein. Specific activity is the ratio of units to protein content in mg. Okay, this is literally the definition of specific activity. sorry, this is a slightly repetitive question. The ratio of units to protein content. A solution of pure protein has specific activity of 5,000 units per mg. If total activity is 20,000 units, how many mg of protein are present? Right? Simple. We have total activity 20,000 units. If we divide that by specific activity, right? This is units. This is units per mg, right? Units cancel. So we'll get the answer in mg. 20,000 by 5,000. The answer will be 4 mg of protein was present in the first place. Which of the following amino acids is negatively charged at neutral pH? Okay. For answer this question, you do need to know the structures of the amino acids. But from this group, there are actually two major amino acids which are negatively charged at neutral pH. One is glutamate, which we saw in one of the previous questions, and the other is aspartate. Which of the following amino acids may bind to an anion exchanger at neutral pH? Okay, this is basically the last question. It is asking for the same information. To bind to an anion exchanger, our amino acid should have a negative charge. So it is asking which of these amino acids will be negatively charged at a neutral pH. Answer to this is again aspartate. Glycine, alanine, serine are not going to carry any charge at neutral pH. In a protein, which amino acid bears a side chain that may bind to a cation exchanger at neutral pH? This is the opposite of the last question. What is going to bind to a cation exchanger? Something which carries a positive charge. So they are asking which amino acid will be positively charged at neutral pH. From this group, the only positively charged amino acid at neutral pH will be lysine. Alanine, methionine, asparagine are not going to carry any charge. So for questions like this, it is very important to look at the structures of amino acids and remember what side groups are there. How does the pKa value of the side chain of an ionizable amino acid compare when the amino acid is free compared to when it is in a polypeptide chain? So for calculation purposes, we state that the pKa value of the side chain is the same, whether the amino acid is in a free form or is in a polypeptide chain. But it might be slightly lower or higher, okay, due to differences in the microenvironment. When the protein is folded and it is exposed to a different environment, it may be slightly lower or higher than its value as a free amino acid. But for calculation purposes, we usually take it as the same. Okay. Indicate at which pH a protein with pI 7.2 would bind most strongly to a column of diethyl aminoethyl cellulose. Okay. We already know DAE cellulose is going to bind to anions, right? Anion exchanger. So our protein should be negatively charged. Now, at which pH will our protein have the strongest negative charge? Okay. The protein will gain more and more negative charge as we move above the pI. Okay. If this is my pI, okay, if I move in this direction, protein is going to gain negative charge. Move in this direction, the protein is going to be more positively charged. Okay. So as the pH increases, the negative charge on the protein will also increase. So the highest pH in this list is pH 9, which is when it will have this most amount of negative charge and bind most strongly to this anion exchange column. Okay. This question I think we already discussed, so I'll skip it. Okay. pKa values of isoleucines, carboxyl, and amino groups are 2.3 and 9.8 respectively. Calculate the isoelectric point. Okay. Now, if you get this question and the name of the amino acid is not mentioned, the correct option would be D. Cannot be calculated without the pKa value of the side chain. Because it is very important to know what the value of the side chain is before we calculate the pKa. But in this question, they have given the amino acid is isoleucine. We already know that isoleucine does not contain an ionizable side chain. Okay? So the only ionizable groups in isoleucine are the 2 amino group and the carboxyl group, and the pKa will be the average of these two, 2.3 plus 9.8 divided by 2, right? Okay, so this will be whatever, 13.1, right? 13.1 divided by 2, right? 2.3 divided by 9, 2.3 plus 9.8 divided by 2 will be 6.05. We'll round that up, call it 6.1. Okay. Which functional group is found on a hydrophobic interaction column? Obviously, it needs to be a hydrophobic amino acid of hydrophobic group. It should not be charged. So the answer to this is butyl. Which of the following best describes the property of globular protein that is exploited in affinity chromatography? Answer is ligand binding. Which would be the best way to separate a protein that binds strongly to its substrate? Answer is affinity chromatography, right? Specific interaction. Which of the following best describes the behavior of proteins in native polyacrylamide gel electrophoresis? Okay, important word in this question is native. Remember, in a native gel, we are not adding SDS, we are not adding any denaturing reagent, right? So the protein is migrating based on its native charge. So migration is going to happen based on its mass to charge ratio. It is going to retain its whatever assembly it has. If it's a dimer, trimer associated with different proteins, that assembly is going to be retained. Okay, because we are not using denaturing conditions in this. Indicate which type of polymer is typically analyzed by polyacrylamide gel electrophoresis in the presence of SDS. Okay. Now, important part of this question is they've added in the presence of SDS. SDS we use when we want to analyze denatured proteins. Okay. Of course, without the SDS, we can also run a protein. It will then become a native gel. Polyacrylamide gels can also be used to analyze DNA. Okay, there's no one is saying you cannot run DNA on a polyacrylamide gel, but because of its pore size, it is more commonly used for proteins. What is the purpose of SDS and SDS page? We just discussed SDS is going to denature the proteins and is going to impart a negative charge, a uniform negative charge to the protein. And because it is imparting a uniform negative charge, right, all of the proteins are carrying the same charge. So any migration that is happening is only based on the difference in its molecular size. Okay? It is not going to do any of these other things. By adding SDS during electrophoresis, it is possible to separate proteins exclusively on the basis of their molecular weight. Electrophoresis of histones, okay, isoelectric point of histone is 8.5, and myoglobin, isoelectric point is 5.5. Under non-denaturing conditions, what will be the result? So first we need to think about what will be the charge on these two proteins. Here, histone pi is 8.5, pH is 7, so pH is under the pi, right? So histone will probably be positively charged. Myoglobin, pi is 5.5, we are running it at pH 7, which is above the pi, so myoglobin will be negatively charged. Since the histone is positively charged, it is going to migrate to the cathode. Myoglobin is going to migrate to the anode. Okay? And it is important that since we did this under non-denaturing conditions, if we did this under SDS conditions, both of these will be negatively charged and both of them will migrate to the anode. Which of the following best describes behavior of proteins in SDS? The best described as migration based on mass. Right? It is not migrating towards the cathode, it is migrating towards the anode. Because SDS is denaturing, it is not going to migrate as a multimer, it is going to migrate as a monomer. Okay? And SDS is imparting an overall negative charge. So the only correct option is C. SDS page, proteins are separated based on their molecular weight. Isoelectric point or pi of an amino acid is blank. We've discussed this extensively. It is the pH at which the amino acid or protein has net charge of 0. At pH corresponding to pi, protein is dash. Okay. At pH corresponding to pi, since it has no overall charge, it is going to be electrophoretically not mobile. Right? In electrophoresis, it is very important that the molecule which you are going to analyze should be charged. Otherwise, it's not going to move in an electric field. So electrophoretically, it is going to be least mobile. It is also going to be least soluble. Okay. An isoelectric focusing gel. Okay. We discussed this technique. It is called IEF. It is the first part of 2D gel electrophoresis, in which we are going to separate proteins based on their isoelectric point. So to separate it based on an isoelectric point, we should have a pH gradient. Right? So isoelectric focusing gel combines pH gradient with an electric field. So basically in an isoelectric focusing gel, what is going to happen is we have a pH gradient, we apply electric field, and proteins will migrate until they reach pH equal to pi, at which point there will no longer be any overall charge on the protein, and so they will not migrate. Okay. What might happen if you forgot to add glycerol and then used it for SDS page? Okay. So the loading dye for SDS page, you will usually contain a denaturing agent. It will contain some dye so you can track the progress of your protein, and it will contain glycerol. Glycerol we add to increase the viscosity of the overall mixture so that it settles inside the well properly. Okay, you're loading the protein like this. What we want is the protein to settle nicely inside the well. Okay. Otherwise, if glycerol is not present, what is going to happen is the protein can start diffusing out of the well. Okay, in the time that it takes you to load the other wells, the protein can diffuse out of your well. So if you don't add glycerol, you will find that it will be difficult for you to load your proteins. Okay? And the samples in adjacent wells may mix. Okay. You ran a Western blot. Okay? Remember, Western blot is when we're going to run an SDS page gel. We're going to transfer the gel onto a membrane, and then we're going to use an antibody against a specific protein of our interest and detect only that protein in the gel. So we ran a Western blot. It is giving 2 bands in each lane. So which are the potential explanations? Okay. So when you think about why should we get multiple forms of our protein, or why are we getting 2 forms with different molecular weights? So what are the ways in which we can get multiple molecular weights? One, there can be some post-translation modification of the protein. Okay, if my protein is getting glycosylated after translation, then maybe I have some glycosylated protein and some non-glycosylated protein. Okay, non-glycosylated protein will obviously have less molecular weight. My glycosylated protein will have higher molecular weight. So this is one explanation. Now, not all the bands we get in Western are specific bands. Okay, of course, we want a primary antibody that will bind only to our protein. But it is possible that there is some other protein in our cell which can react with our antibody. So maybe one of the bands is due to non-specific binding of our antibody. Maybe. We don't know. You have used the wrong secondary antibody. Okay, this is unlikely. I will say this is not a correct answer for this question. Because if we had used the wrong secondary antibody, we would not see any bands. Right? Because secondary antibody does need to bind to the primary antibody to give us a signal. So if we've used the wrong secondary, we are not going to see any bands. You forgot to add bromophenol blue. If you forget to add bromophenol blue, what will happen is we cannot track the protein movement when we run our SDSPH gel. But it is not going to affect the Western blot in this way. Antibody concentration is too low. Antibody concentration is too low, we're going to get the opposite problem, where we're not going to see any bands. Right? We're not going to detect anything. And I would say the most likely explanation for this is there are two isoforms of your target protein. Okay? There are two isoforms which have slightly different molecular weight, and the antibody is detecting both of your isoforms. What happens if you use primary antibodies at too high a concentration? So, obviously we need some minimum amount of antibody concentration so it can go and bind to our protein of interest. And normally, since antibodies are very expensive, we will use them at a very low concentration in the first place. But sometimes in the optimization procedure, we don't know what is the correct concentration. So if we use too much of antibody, what will happen is that we will get increased non-specific binding. Okay? It can go and bind to other proteins which may not be of our interest because it has some low affinity for those proteins. So to prevent this, it is important that we optimize the antibody concentration. You have just bought a new antibody. Which of the following options for samples would help you determine which band on your Western blot is real protein compared to non-specific bands? Right? Like we just discussed, if the antibody concentration is too high, we can get non-specific bands also. So if I have like 3 bands, then how should I know that which one is my protein and which of these are non-specific? So we have here are some options. Sample where the expression of your protein of interest has been knocked down. Okay, this can work. Right? I have a sample with protein expression, sample without protein expression. In my sample with protein expression, I am getting 3 bands. But here, this protein expression has been knocked down. So here, I should not get the band of my protein. So I'm getting these 2 bands, but I'm not getting this band, which means that these two are non-specific bands, and this band is the real band which is corresponding to the protein of my interest. Okay, sure. So this will be a good sample for this question. Probing with secondary antibody only, it's not going to work. It is not going to detect a protein. Loading dilution series of your protein sample. This can also work. Right? If I have a dilution series, then one band should get stronger. Right? As the concentration of my protein of interest increases, the band should get stronger. One caveat is that if the non-specific band is also increasing, then it is possible that as we increase the amount of the protein sample, the non-specific band can also increase in intensity. Okay, so probably the best uh, a condition to test this would be here a sample where the protein of interest has been knocked out. What are ELISA plates with antigen coated wells used to measure? Okay, ELISA, we have discussed, it stands for enzyme linked immunosorbent assay, and we usually use it to test for antibody interactions against an antigen of our interest. Right? So it is usually performed in a 96 well plate where we coat either the antibody or the antigen on the plate and we add the interacting partner, right? And then we detect the amount of binding. So if the plates are coated in antigen, what we're going to detect is the level of antibodies to the antigen that are present in solution. Okay, which of these is not an assay to determine protein content? Answer this is lamellae. Lamellae is actually the name of the buffer system used for STS page. Okay, these are all protein detection assays. They're all very sensitive, or rather, BCA is the most sensitive. These are all fairly sensitive assays to determine protein content. You want to run 7.5% acrylamide gel. Okay. You need to make up 10 ml of gel solution from 30% stock bottle. Okay. So what volume do we need? To do this, you need to know one of the most basic formulas you'll use in a lab, which is C1V1 is equal to C2V2. Okay, concentration 1 into volume 1 is equal to concentration 2 into volume 2. The final sample we need is 7.5% into 10 ml, but our stock is 30%. And we want to know what is V1. Okay, the answer is 7.5 into 10 divided by 30. Right? So you have 7.5, sorry, 75 divided by 30. Okay, answer will be 0.25 ml, which is the same as 250 microliters. Right? No, wait. 7. Yeah, 75 by 30. 75 by 30. So answer will be 2.5. Sorry. Answer will be 2.5 ml. You need to make up 10% ammonium persulfate solution. What mass of powder will you need to make up 5 ml of solution? Okay. You just know what's the definition of percentage. Definition of percentage is 1 gram per 100 ml. Okay. So a 10% solution will be 10 grams per 100 ml. And so if you need to make up 5 ml of solution, the mass that we will need is 10 into 0.05, which will be equal to 0.5 grams or 500 mg. Which of the following statements about SDS page is correct? Separates on basis of size, this is correct. Solubilized but not denatured, this is wrong. Separates on basis of charge, this is wrong. Enzymes retain the biological activities, this is wrong. Only correct statement is that it separates on the basis of size. Okay, this is an interesting question. We've bought an antibody. Datasheet says that the antibody works for immunoprecipitation, but not for Western immunoblotting. Can we predict what type of antibodies they are? Okay. So, can you think of a case where what is the difference between immunoprecipitation and Western blotting? Okay, Western blotting, let us assume standard case, we've run an SDS page gel and then we're going to do a Western. So the major difference is that in immunoprecipitation, we are taking basically whatever cell lysate, okay, or some protein sample, and then we are going to add the antibody to that. So the protein is in its native form. In a Western, since we've already run probably an SDS page gel, we have already denatured the protein. Okay? So if our antibody is reacting to some three-dimensional epitope in the protein, that epitope is going to be lost when we denature it. Right? So uh, obviously, if our antibody is raised against the native protein, there is a chance that it will not react with the denatured protein. So an antibody raised against native protein, okay, or purified protein is not going to react. It may not react in a Western blot. Which of the following hypotheses would sandwich ELISA be a good choice? Okay, sandwich ELISA is basically when we have an antibody already coated on, or we have something already coated on the well, we are going to add a protein of our interest, which is going to bind here, and then we are going to probe it with something else. Okay. So in which of these cases would sandwich ELISA be a good choice? So here you can see it is good when we want to look at interacting partners of a protein. So we can coat the interacting partner on the well, right? We can then add our protein and then detect how much of our protein has bound, right? So if we want to look at the interaction of two proteins, such as protein KRN forms complex with LDT, right? So this makes sense. We can coat LDT on the well, right? LDT will be here. We will add KRN. KRN will bind to the LDT, maybe, right? I have a hypothesis that it is binding, but it may not be the case. And then we will add an antibody against KRN, and that is going to detect how much of KRN has bound. So this is probably going to be a good choice for sandwich ELISA. mRNA binds to 3' UTR. We can also try to do this. Coat the miRNA on the well, add ZIP4, whatever ZIP4 protein is, and then look for interaction, or measure the amount of ZIP4 which is now bound in the well. Okay. So this is a picture for Western blot. Here we can see we have bands in 4 wells, but the bands are of different sizes. And you will also see that the bands have started to spread. Right? There's some spreading of the band. So the question is, we have prepared protein extracts from 4 different tissue types, and then we got the blot above. According to antibody datasheet, we should get band at 130 kDa, right? We should get band here. So what is this? So here we can see that basically the protein size has increased in these tissue types, where here it is 130 kDa. Now, we already discussed that one thing that can change the position of the band in a gel is if we have post-translational modifications. Now, one of these is phosphorylation, but phosphorylation is just adding a phosphate group. It is not going to change the size of a protein very much. Okay, so the more likely explanation is that protein is differentially glycosylated in different tissues. And because of some large glycosylation that is being added here, the size is increased. Here the question is, match the buffer to the protein. So protein A is epidermal growth factor. Protein B is integrin. P4, some transmembrane protein. Protein C is keratin 114, which is an insoluble cytoskeleton protein. And protein D is SOX9. Okay. So buffer 1 is 6 molar urea, 1% STS. This is a denaturing buffer, and one that we will usually use for insoluble proteins. Okay, so this would probably be a good choice for protein C. Okay, so if our protein is going to be stuck in an inclusion body or something, we will have to denature it and then resolubilize it or refold it. So we will usually use a buffer like this, 6 molar urea, 1% STS. So buffer 2 is 50 millimolar tris, 1% Triton X, 5% sodium deoxycholate, and NaCl. Now, we already know that we already discussed that Triton X is what we can use for integral membrane proteins because it is going to solubilize the protein. Right? So for B, we should select buffer number 2. Then the other buffer 3 is 50 molar tris, pH 7.5. This is a very standard buffer. This we can use for, we would usually use in combination with NaCl, but we would use this usually to extract cytoplasmic proteins. So this would probably be a good choice for protein A. So C we're saying buffer 1, B we're saying buffer 2, A we're saying buffer 3. So this is probably the best match of the buffers and the protein. Okay. It's a very long experiment and we're running a bit short on time. Okay, but anyway, we can go through this quickly. You're planning an experiment to determine relative expression between 10 different cell types of a newly discovered protein of some estimated size 20 kilodaltons. How can we set up this experiment? Okay, we want to determine that we have 10 cell types. We want to see which is expressing our protein the most. Okay, so just without going through this option, we can discuss orally what would be the best way. So if you want to determine how much protein is being produced, it is very important to normalize the amount of sample that we are loading in our gel. Obviously, since we want to look at protein expression, we will do a Western blot. It's the easiest way to do this. But first, we should trypsinize the cells, right? Count different cell types, make total protein extract, correct? Since we've already counted the different cell types, we can add different volumes of lysis buffer to normalize for number of cells. Okay. Run on an SDS page gel, transfer to PVDF membrane, use sponsor S, probe primary, probe secondary, quantify band densitometry. Okay, so this is a standard Western blot procedure. We have just normalized the number of cells because we want to compare the relative expression between different cell lines. Okay, so this sounds fairly sensible to me. Okay. The ratio of absorption at 260 to 280 is commonly used to assess DASH. Okay. What you should know is at 260 nanometers, nucleotides have greater absorbance. Okay, and at 280 nanometers, proteins have greater absorbance. And so the ratio of these two is commonly used to assess the purity of DNA and RNA with respect to protein. Okay. Okay, this is a question relating to absorbance, more specifically to Beer-Lambert law, which we use to relate absorbance and concentration. Okay. You measure the absorbance at 260 nanometers of your dsDNA sample and get reading of 0.15. What is concentration of your sample? Okay. 1 centimeter is the standard cuvette length. Okay. So because this is the standard cuvette length, we will take these values of epsilon, okay, it's absorption coefficient, as it is. So A is epsilon c lambda. So A by epsilon lambda will give us C, right? So we need to check what is our sample, double stranded DNA. For double stranded DNA, we have epsilon value of 0.02, so 0.15 divided by 0.02 will give us the concentration of our DNA, 0.15. Okay, which is technically giving us a value of technically giving us a value of 7.5 mg per ml. Yeah. Okay, which is not part of these options, but the correct option to this question would be 7.5 micrograms per ml. You're preparing an agarose gel to run your PCR product. Products are 800 base pairs and 1.2 kb. Okay. Now, uh, depending on the size of the product we want to run, we will select the percentage of the agarose gel. You will see that these products are quite small. Okay. So since these products are quite small, we should have a smaller pore size so that we can separate them effectively. So ideally, we should use a higher percentage of agarose, so the pore size of the gel will be smaller. So it would be appropriate for this to run maybe 1% or 1.5% gel. Let's run a 1% gel. Yeah. So if you want to run a 1% gel, then for 50 ml of gel, we should have 0.5 grams of agarose. Okay. If you're running a plasmid, we might make a 0.8% gel instead. Okay, because the plasmid is bigger. And so if you want to resolve that properly, you should have a bigger pore size. Which of these dyes could you use to visualize DNA run on an agarose gel? Yeah. DNA is already run, you need to visualize it. So we need something which is going to interact with the DNA. The only dye over here is ethidium bromide, which is a fluorescent dye, which is going to intercalate with the DNA. Okay. What additional information could we get from a northern blot that we cannot get using RT-PCR or quantitative PCR? So these are all techniques to determine the amount of mRNA present in our sample. Right? In the northern blot, first we are going to run the mRNA on a gel, then we're going to transfer it to a membrane and then probe it using an oligonucleotide probe. RT-PCR or qPCR, we are going to design, we're going to take the mRNA, we're going to synthesize a complementary DNA strand, and then using primers specific for our gene, we're going to amplify it, okay, and basically track the amplification to get an idea of how much of the template was present. So you may think that both of these will give us the same information, but no. So in the northern blot, what we can in northern blot what we can do is actually see if there are different isoforms of the same DNA, right? Because when we do a qPCR, we are not looking at the size of the product which is formed, we are just looking at the amount of product which is formed. So if we are getting products of two different sizes, it doesn't matter actually in RT-PCR. But in a northern blot, we can see these two as different bands in the gel, so we can determine the relative abundance of splice isoforms with alternative exons. Okay, we have set up a PCR testing for the presence of a transgene in a mouse line. Now, using primers, we have used it successfully, so we know the primers are working. We have the following samples. Okay. Lane 1, we're going to run plasmid. Lane 2, there is no DNA. 3, 4, 5, 6 is our experimental samples. Okay. When we run the samples on the gel, we have band in lane 1, no band in lane 2, and there is no band also in all of these lanes corresponding to our experimental sample. Okay. So uh, since we are seeing a band in one, so this is basically a positive control, this is a negative control, and this is our test. So since we're getting a band in a positive control, we know that the machine is working, right? We know our procedure is working. So there was no problem in running the gel, there was no problem in the PCR, and the primers did not degrade. So now, since you are not getting any band here, one option is that the 4 samples don't contain the trans gene. That is possible. Or the other option is that the mouse DNA that we isolate is of poor quality. This is also possible. If the template is of poor quality, then we will not get effective amplification. Okay, and we will not see any band in our PCR reaction. Okay. It's another problem on agarose gels and PCR. In the figure above, the large boxes indicate exons with the size indicated within the box. Okay. So there's exons in between are introns, so these will get spliced out. Red arrows indicate where the PCR primers bind. So PCR primers are binding here at the beginning of X1 and here at the beginning of at the end of X5. Okay. Positive control is a plasmid that has X1, X2, X3, X4, X5 and no intronic sequence. So positive control is basically the sum of all this, which is around 500 base pairs. Sample cDNA is present in the cell line, okay, where we're getting the same band as the positive control, which is a full length gene, but we're also getting these lower bands. So since we're getting these 2 lower bands, what that basically indicates is that some of these regions in between are getting spliced out, right? So if we lose these regions and these 2 are spliced together, obviously we'll get a product which is around 113 base pairs, 213 base pair, okay, which is going to come around here, right? So the obvious explanation is that these are alternative splice isoforms, X1, 2, 4, 5 upper band and X4, 5 are lower band. Obvious explanation. Very basic laboratory question. Which of these pipettes is said to dispense 32.7 microliters? Yeah, so first let us look at the pipettes themselves. So here, when we look at the pipette display, this is going to be the highest digit under the highest count of the pipette. So if this is a P1000, that means it dispenses 1000 ml. So this is going to refer to the hundreds point. So this will normally, or rather this is going to refer to the thousands. Okay? So for P1000, okay, the display is going to go 1, 0, and then you have the variable. So this is 1000. On a P100, this upper unit is going to refer to the hundreds place. Okay? So same deal. This will probably refer to
100. Okay. Um, when we look at 32.7 microliters, this is going to be typically under the least count of of a 1,000 microliter pipette. Okay, 1,000 will usually dispense 1,000 to 100 microliters properly. Okay, so in a uh, in a 100 microliter pipette, this is 0, 32.7. So this is actually set at 32.7. This is set at 327. Okay, and this has a least count of P10, so this is set at 3.27. Okay, this is set at also set at 327. So the only pipette set at 32.7 is actually B. You're about to thaw a vial of cells. What hazards are associated with retrieving the cells from liquid nitrogen? As we know, liquid nitrogen is extremely cold, and if it comes in contact with your skin, it can actually burn it. But also, since it is liquid nitrogen, it can sublime to form nitrogen gas, okay, which can also pose an asphyxiation hazard. So both of these are true. Why should you be extra careful using Ethereum bromide? So Ethereum bromide we know intercalates with DNA, so it can also intercalate with your DNA, right? So it can cause DNA damage. When you leave a lab, what happens to your lab book? Your lab book will contain the record of all the experiments you've carried out in the lab. So it has to stay in the lab itself because it forms a reference point for everyone else who is going to work in the lab. Okay, so it remains with the lab or supervisor. What specific additional precautions should you take when using the substance with this label on it? So this label you see a flame surrounding an O. So this is a, a symbol for oxidizers. Okay? If the symbol was just a flame like this is a symbol for flammable reagents, but because it has an O, it is an oxidizer, so it will feed a flame. So obviously, if we're going to use this, we should keep it away from flammable material. Right? What does this symbol mean? So this exclamation mark means that it is means caution. Okay, it means that it can cause damage to your eyes, skin, and nervous system. Okay, bought a new reagent. On the datasheet it says stable at 4 degrees for 1 month, long-term storage for 1 year, and avoid repeated freeze thaw. You don't think you will use the whole vial in the next month, what should you do? So if you get reagents which are unstable at 4 degrees, and also you don't want to repeatedly freeze through it, the best way to deal with this situation is to make aliquots of the reagent and store it in the deep freezer. And so when you need to perform an experiment, you can take out one aliquot and thaw only that aliquot. So the rest of the reagent is remaining frozen. So the best way would be to calculate how much you will need for each experiment. So for one experiment, you can just pick up one tube, aliquot it, and then store in a labeled container. What temperature should the test solution for your pH measurement be to be accurate? Okay. So most temperatures most pH meters will come with a temperature probe because pH is actually de- changes depending on the temperature of your solution. Okay. But if there is no temperature probe, then it should be at the same temperature as the buffers used to calibrate the meter. In flow cytometry, what is side scatter a measure of? We discussed that In flow cytometry, we get forward scatter, which is a measure of the size of your cell. Right? And we also get side scatter, right, which is from the internal environment of the cell, and the amount of scattering is based on the granularity of the cell. What you have to adjust to ensure that fluorescence from one channel does not spill in over into other channels. So, because the excitation and emission wavelength spectra of fluorophores do overlap slightly, we need to adjust a parameter called compensation, okay, which makes sure that the two channels are separated from each other properly. What information do you need to know when choosing fluorophores to use in flow cytometry? So what we should know is obviously excitation and emission spectra of the fluorophores, and we should also know which lasers and filters are available on the flow cytometer, right? Because there's no point in choosing a fluorophore which we cannot then detect in a flow cytometer. We are performing a flow cytometry experiment with a previously characterized antibody on a new cell type. Which of the following controls would tell you if your new cells have autofluorescence? So autofluorescence can be a property of some uh, components within the cell itself. Right? So even without a fluorophore, you will get some background in the channel. Okay, so to know if a cell has autofluorescence, we should run a sample with our cells, no antibody at all. Okay. We can also try cells processed with a secondary antibody only. Okay. Or cells process isotype matched primary antibody and secondary antibody to the primary. This is the most stringent control, okay, because uh, this is basically exactly the same as your test sample, except that the primary antibody is not going to bind to your protein. So if you have autofluorescence, this is the most stringent control that we can use. So these are some examples of scatter plots from a flow cytometry experiment. We have probed the cells with two different antibodies, one labeled in red and one labeled in green. So red is on the x-axis, green is on y-axis. Plot 2 is negative control. Okay. Which plot would indicate that the subpopulation of cells expressed green protein but not the red protein? So this is negative control. Okay. So this group is going to be present in our sample. But we are expecting that there will be some expression of green but no expression in the red, right? So basically our population should move along the y-axis, but it should not move along the x-axis, but it should not move along the y-axis because there is no expression in the red. So the plot which is most closely matching that is this plot 1, right? So you can see the population has moved. We're getting this population here which is slightly further along the x-axis, but it is at the same height on the y-axis. So the red protein is not expressed. So plot 1 is the most appropriate answer here. Okay. Here we have the same cells, but this time we have tried permeabilizing the cells. In permeabilizing, what is basically happening is we're making little holes in the cell membrane, which allows the antibody to enter the cell, okay, and bind to targets which are inside the cell. Okay. After permeabilizing, what we see is that basically the population is shifted upwards, right? It's shifted along the y-axis, which means the red fluorescence has increased. But the green fluorescence has not changed, right? Because it's not shifted along the x-axis, it's just shifted up. And so what this means is that the red, whatever this red fluorophore was interacting with, is probably a target which was inside the cell. Whereas the green target, which is not changed after permeabilization, was probably present on the cell surface. So, antigens recognized by the red antibody are intracellular, antigens recognized by the green antibody on the cell surface. Right? Exactly what we discussed. Test which of the following hypothesis would transmission electron microscopy be an appropriate imaging modality? Right? Transmission electron microscopy, remember, will give you basically an image of the uh, interior of your sample. Okay, surface scanning electron microscopy will give you an idea of the surface. It's to give you an image of the sample. So when we want to look at, say, a different cell uh, differences in, say, cell organelles or something like that, transmission electron microscopy might be an appropriate choice. So of these, I think the best option for TEM here would be last option here. The inner plaque of hemidesmosomes does not assemble, right? So here we would have to produce two samples, uh, mouse epithelial cells with kindlin 1, mouse epithelial cells without kindlin 1, do TEM on both of them, and check whether this inner plaque of the hemidesmosome is present or absent. Right? Which is something that we should be able to see in TEM quite easily. You're comparing two different cover slips that have been fixed and processed for indirect immunofluorescence microscopy with the same antibodies. What can we determine? Okay, we can determine things like relative protein location. We can determine relative protein quantification. Obviously, we can see that fluorescence might be higher for one, higher in the other. So we can determine both protein location and quantification. Okay. So these are the options of fluorophores that we have for our experiment. Okay. We have 4 fluorophores with this kind of excitation and emission spectrum. Then we need to know which microscope will work for this experiment. Obviously, we get some spectral overlap, right? So, but we can avoid, so we can sort of separate this into different channels by using different filters. Since we want to get a fairly good resolution to check relative distribution of 4 proteins, so we need to get fairly good resolution, we will not use an epifluorescent microscope, we will use a confocal microscope. Then when we look at the excitation spectra, and we see which of these is most appropriate for excitation. We need 405 to excite this. So 405 is kind of lowest, will excite this fluorophore. 488 for the green fluorophore. This will need somewhere little under 550. This will need somewhere near 650. So this matches most appropriate laser lines that will be required for the excitation. Right? See, if we have the major difference between these two is present at the 720 nanometer laser line. But 720 laser line would come here, right? And that is outside the excitation spectrum of this last fluorophore. So we will not be able to excite this fluorophore using this setup. So the appropriate setup for these is this last one. Okay. Another question using the same spectrum. Fluorescence resonance energy transfer uses light emitted from one fluorophore to excite the next fluorophore. Which of the following would be the best FRET pair? So basically, for something to become a FRET pair, there should be a large overlap between the emission spectrum of one fluorophore and the excitation spectrum of the other. Because the light emitted by this is basically going to excite the other fluorophore. So if you look, so if we look at which of these spectra have the greatest overlap, okay, so we can see that
this green emission spectrum and this red excitation spectrum have quite a bit of overlap, right? This is all the overlapping region. So if I excite this green fluorophore, it will emit this light, which will be able to excite the red fluorophore. Right? So the most appropriate FRET pair over here, because if you look at the other pairs, this is the overlap and this is the overlap with this pair. So this is the best pair over here. So if you have donor green and acceptor red, it can act as a FRET pair. Okay. Which objective would allow you to obtain the highest resolution images? Okay. So here we have given images of 4 objectives, all of which have the same magnification. Okay, these are all 60x objectives. But here you will see another value, right? This is what we call the numerical aperture, right? Obviously, as the numerical aperture increases, the resolution of the image that we can obtain will increase. And so we will look for the objective with the highest numerical aperture, which this is 0.95, this is 0.7, this is 0.14, 1.4. This is an oil immersion objective. So this will give us a higher resolution image than these two. This last objective is 1.49. It's also an oil immersion objective. This is getting pretty close to the theoretical limit of the numerical aperture we can achieve. So since this has the highest numerical aperture, this will give us the highest resolution image in this set. Okay, so we've gotten this image from a microscope, but you can see that this image is not illuminated properly, right? We just have these spots of yellow light, but the rest of the image is mostly dark. So our images have issues with uneven illumination. Which part of the microscope should we adjust? Okay. So for this, we need to know which part of the microscope is responsible for controlling the illumination of our sample. Okay. And the answer to that is the condenser. Okay. The condenser is responsible for making sure that our sample is evenly illuminated. The diaphragm and light source will basically control the amount of light which is falling on a sample, but the illumination of a sample is controlled by the condenser. Okay, condenser, the showing is here. Okay, what is photobleaching? So this is a concept that comes when we talk about fluorescence. If we expose our fluorescent sample for a long time, it will start to lose its fluorescence. Okay? And what is happening is that light energy reacts with the fluorophore and causes permanent structural damage. And because the fluorophore is getting structural damage, it loses fluorescence with time. Okay, so video is not given, but we have epithelial cells which have been induced to express some protein paxillin along with GFP tag. Okay, so we have paxillin with GFP. So obviously the protein is already tagged with the fluorophore. So what are the advantages and disadvantages of this approach in comparison with using antibody staining? Right, so if you were going to do a traditional fluorescence microscopy, you would just have this protein. You would have some antibody against this protein, and the antibody will be tagged with the fluorophore. So we are going to compare these two approaches. Okay. So disadvantage. What are the advantages of this technique? So why would we want to do this? Right? So since the protein is being tagged with the fluorophore while it is being produced, we can look at the expression of the protein at different times. Okay? And we can identify how the protein is moving inside a live cell. So one advantage is that we can answer questions relating to protein dynamics, that is comparing focal contact assembly or turnover rates. Right? So we can visualize the protein at different time points and see how it's working. However, one disadvantage is that this is a disadvantage of any tag that you can add to a protein. The fluorescent tag or expression system can affect the localization and function. This is a disadvantage for any his tag, strep tag, GFP, any tag that you're adding to your protein. Okay. The biosafety level that is recommended while working with Mycobacterium tuberculosis is dash. So, Mycobacterium tuberculosis uh, poses a great danger to the person who's working, and it can spread through aerosol contact, and so it is a biosafety level 3 pathogen. Okay, the parameter to define binding capacity of an antibody is dash. Answer to this is avidity. You will be aware that antibody binding is characterized by two concepts, avidity and affinity. Avidity is basically telling you about the total binding capacity of an antibody, whereas affinity is measuring the strength of each individual interaction that the antibody is making. Direct agglutination assay is used in DASH. So, direct agglutination assay is when we have a sample which has some antigen. We are going to add a very high concentration of antibodies, and these antibodies are going to cross link. Okay, so we have maybe a polyclonal antibody which is going to cross link the different antigens and form a precipitate. And this is used commonly in ABO blood typing. Okay, we call this hemagglutination because the RBCs get cross-linked in ABO blood typing, and then they precipitate out of the solution, and you can see like a haziness in the sample when it is positive. Okay, what happens to the color of mammalian cell DMEM media when the pH of the media decreases? Now we know that DMEM media and most mammalian cell culture media contain a pH indicator called uh, phenol red. And phenol red is red in color at neutral pH, but as the pH of the media decreases, the color of phenol red will change towards yellow. Okay, as the pH of the media increases, okay, as it goes above 7, the color will become more pinkish. The reagent used to develop HRP's based secondary antibody system in Western blot is luminol H2OT. Okay, so HRP is horseradish peroxidase, which requires peroxide as a substrate, which is H2OT. Function of phenol red in cell medium is as a pH indicator dye, as we just discussed. Philippine staining can be used to study phagocytosis of latex beads by specific cells. Philippine stains the lipid. Okay, it's basically stains the lipid bilayer, and you can then see the phagocytosis because you can see it move around to engulf the latex bead. Which of the following proteins prevents reannealing of ssDNA? So, when we have separated the two strands, okay, during DNA replication, the two strands are kept separated due to the binding of a protein called single-stranded binding protein, which is going to keep the two single-stranded DNA separate. Number of reference electrodes present in a pH meter is dash. pH meter will typically have one reference electrode. How many grams of sucrose will be required to make 100 ml of 0.1 molar sucrose solution? Molar weight of sucrose is 280. Okay. Let's start from the basic. What is the definition of molarity? Okay. Molarity is 1. So if I have a 1 molar solution, 1 mole of the mixture in 1 liter. Okay. But if we know what is the molecular weight of is basically grams per mole, right? So I know that 1 mole of sucrose has a weight of 280 grams. But what are they asking me to make? They're asking me to make 1.1 molar sucrose solution. So 0.1 molar will be equal to question mark weight 100 ml, which is the same as 0.1 liters, right? So uh, to find out the weight of sucrose required, we will have to do 0.1 molar into 0.1 liters into the known molecular weight, 280, which basically will give us 2 decimal point, 1 decimal point here, 1 decimal point here. You will need 2.8 grams of sucrose. Research fellow is facing the problem of acrylamide polymerization even when he is allowing the reaction to stand for 30 minutes. Meaning he has made the full reaction, he has kept it at room temperature for 30 minutes, but it's still not polymerizing. So which component is most likely to be missing from the cocktail? Now we know that the polymerization of acrylamide requires the presence of free radicals, which we are providing with ammonium persulfate, and the reaction is catalyzed by the presence of TMAD. So if we have added the acrylamide and it's not polymerizing, most likely one of these two components is missing. Uh, from this list, TMAD is acting as the catalyst, so if TMAD is absent, it will not polymerize. Okay. Complex protein mixture separated by SDS page, and the RF value is calculated for 4 protein brands. Okay. So basically SDS page, this is going to say this is our dye front, and RF is basically the distance traveled by the protein divided by the distance traveled by the dye front. So obviously if the protein is closer to the dye front, the RF value will be more. The protein is further away from the dye front, RF value will be less. Right? So this is distance traveled by the dye. This is my protein. This is the distance traveled by the protein. So the protein band closest to the tracking dye would be the protein band with the highest RF value. So here if you arrange it in order, it will be D, B, A, C, right? Because C has the lowest RF value. So the one which is traveled furthest is protein D. Agarose acrylamide composite gels are used to analyze DASH. So we know agarose gels are usually used to analyze nucleic acids. Acrylamide gels are usually used to analyze proteins. Normally we use composite gels to analyze large multimeric protein complexes. Okay, because the pore size of agarose is typically bigger than acrylamide gel. So if you have a large protein complex which you cannot run or resolve properly in an acrylamide gel, we will typically run it in a composite gel. Nuclear protein is involved in the regulation of gene A. Protein was incubated with gene A and run in the gel filtration column, which is the correct statement in the following. So here we have protein, to which we have added some protein A, which is its ligand. So now, obviously, we can see that the molecular mass, apparent molecular size of this complex has increased. So, as it is increased, obviously the hydrodynamic surface area has increased, so this is wrong. The peak corresponding to the protein ligand complex will show a right shift, this is wrong, right? As you know, gel filtration chromatography, as elution volume, the proteins elute in decreasing size, in order of decreasing size. So if this is my protein without the ligand, once we add the ligand, because the size is increased, the peak will shift towards the left. Right? KD value is not of any consequence in this experiment as such. The protein ligand complex will have lesser retention time is true, because it is bigger, it is going to elute first from the column. This is true. Okay, I think it is 8 o'clock, so I will call this session to an end. Okay, there are a number of other questions which are also present in this PPT, so I'll upload that to the this folder which is accessible. a drive folder which is accessible. There are a lot of other questions that you can go through and solve for your practice. Okay, so these are all questions taken from previous year's GATE papers, previous year's CSI papers. So these are the kind of questions you will see in competitive exams, and they're all related to the concepts that we have discussed in this course. So as always, I think this will mark the end of these problem solving sessions. But if you do have any other doubts, please feel free to use the question and answer tab in the course announcement section. Okay, and thank you for your interest.